Good morning, good afternoon, good evening, children. Welcome to module number 3 of under genetics. We are going to learn about DNA replication here. We are going to learn how are the genes located in a chromosome. Before that, let us revise DNA structure with a song. Enjoy it.
What is a nucleoside? What is a nucleotide? And what is a nucleosome? Let us see. Nucleoside has a base in sugar. Nucleotide has a phosphate too, but no histone. But no histone. Nucleosome is a big brother. Oh yeah. He has many nucleotides with him, 8 histones, and a lot of DNA. The DNA wound round 8 histones. 8 histones. That is what a nucleosome is.
I'm sure that you enjoy the song. Create a song on nitrogenous bases and share the lyrics with me. Moving on, in the double helix of DNA, there are two strands, and both the strands show definite orientation. What do I mean by definite orientation? See, in a DNA strand, there are two ends. One end, which is having the sugar molecule, which is called the 3' end, and the other end, which is having the phosphate group, which is called as the 5' end. If strand 1 in a DNA molecule is having 3' end at the lower end and 5' end at the upper end, then strand 2 will have the 3' end at the upper part and the 5' end at the lower part. So, both the strands run anti-parallel. You can see it in the diagram. So, children, after knowing so much about DNA, I'm sure there may be 100 questions in your mind. If I have 46 chromosomes in my body, where are the genes of my height or where are the genes which control my eye color located in these chromosomes? There are so many other doubts in your mind. All these needs a lot of time to be cleared, but in the next few modules, basic understanding of these will be gained by you. Before I proceed, I would like to tell you there is more than 3 billion nucleotides in each cell of our body, in the chromosomes of our body. Some of them have DNA, which code for proteins. Some of them have nucleotides, which do not code for proteins. Research is going on to exactly determine what their role is. There is a lot of scope in this branch of biology. If you are interested, it is the right direction for you. So, before we proceed to replication, a brief recap about DNA. It's a polynucleotide, it's a macromolecule, it has a double stranded structure, it has a helical structure, and above all, it can replicate itself. So, let us go to the replication part. So, for the replication of DNA, the two strands in the double helix, they have to unwind, and after unwinding they have to separate from each other, and then the parallel strands have to form. So when the replication starts, the two strands start unwinding and separating from one of the ends of the double stranded DNA. As soon as the separation starts from one end, the new parallel strands start forming. Now, how do these two strands separate? By breaking off the hydrogen bonds between the nitrogenous bases. So, which is the exposed area of the DNA strand? It is the nitrogenous bases. So, parallel strand will form pairing with those nitrogenous bases, those who are pairs of the existing nitrogenous bases. So the new strand starts forming next to the original strand towards the nitrogenous bases. Now, as the formation of the new strand progresses from one side, the other end of the DNA slowly gets unwinded, separated, and the duplication process continues from the previous end. So, this replication of DNA will continue till the DNA double helix completely separate from each other and form two double helixes of two strands each. The enzymes helping in this process are helicase, polymerase, ligase, etc., which you will be learning in the higher classes. Even some RNA molecules help in this process. So, once the process is complete, the DNA looks like this, two helixes with double strands each, and you can observe in the diagram that both the helix have one of the DNA strands which was there in the original DNA helix and one new strand each. As it is having one old and one new strand, this method of replication is called semi-conservative method of replication. This method is unique, and even if a single error occurs in this particular mechanism, it's going to make a huge difference in the organism, in the proteins, in the characters, and in everything in the organism. Hello students. When you're copying a sum from the board when your maths teacher has done it, or when you're copying down when a teacher dictates a note, then you may make small little errors in your notebooks. For example, if I check through your notebooks, the same answer might have been dictated by me, but there may be a word extra in one notebook, a letter extra in another notebook, a word left out in a notebook, a full stop there in a notebook, no full stop in another notebook, and so on. All these small little errors are not going to make really any difference in the work you're doing. But if we take the copying mechanism of a DNA molecule, during this copying, even if there is a very, very small error, which amounts to 0.01 percentage, that can make a huge difference in the type of protein formed in that organism. How? The DNA strand with nucleotides, it has been discovered that three consecutive nucleotides code for one particular amino acid. So even if there is one change, one letter, one nucleotide having a different nitrogenous base, the code becomes different. The type of amino acid form becomes different, and the type of protein formed and the structure and the functioning of the cell becomes different. So with an example, let us try to understand how it happens. Here is a hypothetical example of a nucleotide sequence with their bases. We have discussed that three consecutive bases in a DNA strand code for one amino acid. So there are two strands here. Let us take strand 1. The nucleotide sequence is like this. C, from the bottom I am taking C, A, T, then A, C, T, then T, A and G, which is coming on the other side because the stand is twisted. So which are the three consecutive bases here? CAT, cat, which may code for one amino acid. ACT, act, another triplet code which will code for second amino acid. TAG, tag, another triplet code which will code for a third amino acids. So this strand number 1 in this double standard structure will code for amino acid 1, amino acid 2 and amino acid 3. Now in the next slide we will be seeing if there is a change in any one of the codes, what will be the difference. In this you can see that even if one base is changed in a triplet code, it makes a huge difference in the formation of amino acid, in the formation of protein, and in the formation of the character of an organism. Let us see how. Let us think that the sixth base, that is T, gets deleted during DNA replication. Then the sequence will become CAT, which will be coding for amino acid 1, ACT again coding for amino acid 2, but A and G will be left alone without a third nitrogenous base. It cannot code for anything as it is not a triplet code. We can take another situation. The first nitrogenous base here, cytosine C, is by mistake replaced by a thymine when the DNA replication happens. Now the triplet codes are TAT, which may code for a fourth amino acid, not the first amino acid. ACT will code for the second amino acid, and TAG will code for the third amino acid. So instead of the first amino acid, here this particular piece of DNA will code for a fourth amino acid. So the amino acids are changing when the codes, even one error happens in the codes. Such changes are called either deletions, duplications, inversions and so on, which you will be learning in higher classes. These changes will change the amino acid, change the protein and change the characters. Such changes in the nucleotide sequence are called mutation. These mutations are responsible for the coronavirus, which has shaken all of us in the last more than a month. It has shaken the whole world. What is mutation? It is a sudden change in the DNA molecule leading to change in the character of an organism. And how can we define a gene now? A gene is a segment of the DNA which has got codes to control one character in an organism. Or it can also be defined like this. A gene is that sequence of nucleotides in a DNA molecule which code for proteins controlling one character in an organism. These are two important definitions in this lesson. They should be learned. Mutation can be caused due to duplication, deletions, translocations, inversions. All these you will be learning in class 12, and gene is an important concept for class 10. So let us revise with the give reason questions. Question 1. DNA is called a polynucleotide because it's made up of number of nucleotides, not 10 or 100, but thousands and 10,000s of that. Second one, the codes present in our DNA are called triplet code. 3 nitrogenous bases in a DNA code for one amino acid. Because 3 nitrogenous bases are involved, it's called a triplet code. Third one, the replication of DNA is termed semi-conservative. This is because when 2 double helices are formed, each new double helix will have one old strand and one new strand. So it's a semi-conservative method. It is apt to call doubling of DNA as replication and not duplication. Duplication can happen for a bits and pieces of DNA, for a certain segment of DNA. But when we say replication, the whole DNA strand unwinds, becomes the template, and a new strand is synthesized. So it is replication. Differentiate between a gene and a chromosome. Structure-wise, a gene is a segment of DNA controlling one character, whereas chromosome is a condensed and coiled form of a whole DNA molecule containing many genes. Function-wise, gene controls and transmits one character from parent to offspring, whereas chromosomes have many genes in it, so they control and transmit many characters from parents to offspring. Coming to the next difference, third end and fifth end of a DNA strand. The 3' end contains a deoxyribose sugar, that is the type of molecule present, and the 5' end of a DNA strand contains the phosphate group. So these are some of the important questions. Now let us all move to the next group of question, that is diagram based question. So the first question under diagram based question, give a suitable heading to the diagram. The left side of the diagram shows a DNA double helix and the right hand side shows the DNA getting copied. So it is DNA replication. Distinguish between the strand label 1 and 2. Two is the old DNA strand acting as a template, and one is the new DNA strand synthesized. Which are the molecules present in the backbone of the strand? It is deoxyribose sugar and the phosphate group. Full form of letter T. It is thymine, a pyrimidine under nitrogenous basis. With which lettered molecule will it pair with? Adenine, that is the letter is A, and the type of bond between them will be double hydrogen bonds. If A tries to pair with C, what will happen? What is the change? What will it lead to? It will lead to change in nucleotide sequence, and so it will lead to mutations. I hope you understood this module, what all taught in this module. Science is what makes you think, children. There's a lot to understand, there's a lot to discover, there is a lot of excitement ahead. Think science, learn well, watch the video, and keep in touch. Thank you and bye.